There we go. All right. So your exams are all graded. Your exams are all entered. All right. You saw your scores. If you got 70% or below on that exam, come to my office, do the extra credit assignment. All right. You get half the points back that you missed. I've got some time even after lecture today if you happen to have some time. It's really quick. I'm right across the street. I can just give you the materials you need. Question? No. Yeah, it's really meant for folks who fail the exam, to catch up. Yeah. Yes.
It sounded like from the announcement you sent out, you can only avail yourself of this opportunity once for the entire duration of the class.
Yep, and we have 2 midterms. So if you could do it if you get below 70 on the next one, but you got above 70 on this one, you could do it for that one. You know what I mean? And listen, if you finish your second exam and you're like, John, I got 72 on exam 1. Can I please do the extra credit? Okay, I'll let you do it. All right, because the difference between somebody getting 68 and 72 is very minimal, and so I can be flexible there. But yeah, awesome. Hopefully now you see how the exams look like. They're all gonna be like that. Everything comes straight from the study guides. Everything comes from the PowerPoint slides and the reading to try to make it very clear what I want you to review. So just, our next exam is in week 11. All right, we're gonna do it on the Thursday of that week. I've got it all scheduled, I think. That's how I have it scheduled, right? Let's look at it right here. Nope, it's gonna be on the Tuesday of that. Wait, it's gonna be on the Thursday of that week, yes. Okay, cool, good. I just wanted to double check my schedule's laid out like that. To give us time to cover soils, mutualisms, catch up. I'll be very clear about what's available, what's covered on the exam and the study guide, same thing. I keep repeating myself. Are there questions about that? Awesome. Okay, so today we're gonna move into our population ecology stuff, which is gonna be a lot of fun. We finished germination last time. There were a couple of other little pieces at the end of this PowerPoint that we didn't necessarily have time to talk about. Stratification are signals that break. Sorry, yes, please. The final is, but exam 2 is not. And once again, study guide tells you everything you need to cover. So it's all there in the study guide. If you're ever nervous about I know I asked John if it was cumulative or not, but I can't remember, study guide. Yeah. Cool. All right, let's keep moving forward. Stratification, these are cues that break dormancy. These cues can be periods of light, periods of darkness, the presence of water, the presence of heat, the presence of butanolide in smoke, all things that we briefly touched on. But stratification in general is just talking about giving the plant, the seed, cues to break germination. So cold stratification would be the plant gets cold and that breaks its dormancy and allows it to germinate. Okay, cool. Heat, cold, or cold stratification, it's usually about making sure that you don't emerge during the winter. So the cold stratification has to happen in order for the seeds to be ready to germinate. That way, dormancy gets broken in the winter so that in the next spring, when water hits, those seeds can germinate. Sometimes cold stratification works by breaking a seed coat. There's so much overlap in these dormancy mechanisms. It's just ridiculous. All right, so dormancy's complicated. That's my main takeaway from this one. All righty, I hope you enjoyed the dormancy piece. I really like this, so we'll talk about this really briefly. It has to do with seeds being able to detect red versus far red light ratios. The idea is if the seed can tell how much photosynthetically active radiation is actually hitting it, relative to how much total light is hitting it, it can tell how much canopy cover there is. Because photosynthetically active radiation gets filtered out by the canopy, but far red, light doesn't get filtered out by the canopy. So some light passes right through. So if you have very little red light, which gets absorbed by the canopy, but a bunch of far red light, that indicates to the seed, oh, there's canopy closure. I don't want to germinate right now. So it's an important cue, and it's just a cool thing to think about, these red to far red ratios. Humans use the same principle to look at photosynthetic health of plants by blasting them with a bunch of broad spectrum light and then seeing how much of the photosynthetically active radiation gets absorbed. It's called the Normalized Difference Vegetation Index, and it's just fascinating. Don't worry, I'll talk about it more later. Yes? Oh, it depends, right? So every seed, all these seeds have different combinations of germination cues. So this only really makes sense for a seed that's on the surface of the soil, right? And some seeds, most seeds don't have a way to bury themselves. So most seeds germinate on the surface, right? But it's not always the case. So it's a good catch. Yeah. Yeah. So then is this going to work? I mean, it depends. The still light could pass through the leaf until it turns brown and gets all embedded with tannins. The tannins all get produced. Yeah. It's something to think about. Like, how do seed germination rates differ in forests where the leaves fall versus forests where the leaves stay on the trees? It's a great question, a great ecological question, actually. Yes?
That reminds me of kind of what we were talking about with the different characteristics between competitive, for rural, and the third guy.
The stress tolerant.
With the competitive ones having a lot of leaf litter that is not deciduous and is persistent, so that might actually help. Perhaps one of the things that makes them competitive is that it suppresses the ability to
Germinate. Yeah. Yeah.
So the plants quite tricky when they're thinking that there's more canopy cover than there is.
Very solid. I love it. And this is another thing to think about, right? The CSR framework of talking about life history strategies. Well, dormancy is a part of life history. So the CSR categories are going to have a lot of time commonalities in what their dormancies look like. So it's really cool to tie this all together. I support it. I love it. Thank you. All right. Anyways, we're going to just rock through this because I really want to get into our life history stuff. We talked about making the seed coat permeable with abrasion. Remember, stratification is just something that helps to break the dormancy. So yeah, sometimes you got to take a seed if you're trying to get it to germinate and scratch it with a little piece of sandpaper. That's meant to approximate the seed passing through the gut of an organism, usually. Anyway, seed banks. All right, seed banks are just a bunch of dormant seeds in the soil. Some plants make different seeds that are meant to stay dormant different periods of time. You're banking some seeds. It's an investment. Reasonable enough? Awesome. Seed banks, hey, yeah, these seeds can be viable after so long. It's all fascinating. It's all interesting. Great. Jansen-Connell hypothesis. We love it. It's everybody's favorite figure. All right. Are there any questions about it? We've seen it a few times now. Yes?
I spent way too long staring at the two different lines and trying to figure out what the two different lines represented.
Yeah. Yeah, totally. Number of seeds per unit area. Burp. Right here, because you're more likely to distribute closer. The probability of seed dispersal decreases as distance from the parent plant increases. All right, and then the probability that they'll mature. This hypothesis is that that will increase, eventually plateauing.
Okay, so that's the likelihood of the plant survival then?
Yeah, that the seedling will actually survive. So it goes up, and then at a certain point, you get so far away that the probability is the same, But here,
places where there are seeds and the seeds will survive.
Perfect. Thank you. I was trying to figure out a way to summarize it, and that does it. Where there are seeds there, and the seeds will also survive. That's the sweet spot. Cool. I love it. let's go ahead. I don't know. I used to not love talking about this population ecology PowerPoint, but I made some serious adjustments to it. And now I'll tell you, I just can't wait to talk about population ecology with you. Here's the thing, folks. If you're going to continue with ecology, you're probably going to be doing population ecology. Most ecologists don't study individual animals or individual plants. I mean specifically an individual, right? You look at the behavior of a population or the behavior of a community. So population and community ecology is what most ecologists study, and then you work with specialists for the different species that you consider as significant within the community. That kind of makes a little bit of sense, right? And now later on, ecologists will specialize in their careers and be like, I specialize in this population or in this species, so I learn everything about that species, but with a focus of how it interacts with others and how individuals of that species interact. So population ecology can be very different from just the biology of a species or of an individual of a given species, because you're concerned with how it interacts with other individuals of that same species, the environments it tends to inhabit, and the role it plays within that environment, right? Does that kind of sort of make sense why population and community ecology is such a big focus of the ecologist? Yes? Awesome. Okay, cool. Populations, how do we talk about them? In statistics, the phrase population, it just means it's a group that we're trying to, this language, from which we wish to draw inferences, all right? A population is like a subsample, or like a sample, okay? But in ecology, okay, a population's a group of individuals of the same species in the same area and also at the same time. So a population's a group of individuals of the same species that actually interact in some way. Okay? Cool. In genetics, it's a group of interbreeding individuals of the same species. How is that different? Well, check this out. You can have a population, an ecological population, where not all of the individuals actually breed. And if all the individuals don't actually breed, they become reproductively isolated, all right, and they can develop different genetics, and you can get speciation. So from the perspective of the geneticist, it's all about, do the individuals actually breed? So a genetic species concept could say that, hey, there's some mobs over here in this part of the forest, and there's some mobs over here in this part of the forest, and they look a little bit different, and they don't breed with one another. Or there's these two little subpopulations of birds, and these ones, they happen to have a slightly different beak. And they overlap and move between their different areas, but their mating calls are different because of the acoustics of their beaks. So technically, the geneticist would say those are different populations because there's no, gene flow between them, all right? Whereas the population ecologist would say, well, they still carry out the same role in the ecosystem, and they still compete with one another, and they do all of these things. They still require the same resources, all right? So they're a single population still. It's a weird distinction, but defining your terms is always really important, especially when they're big foundational terms like this. Like we talked about the species concept a little bit informally, like what makes something a member of the same species. And that's up for debate. Different fields talk about species differently, and different fields talk about populations differently. But we're doing population ecology. So it's groups of individuals of the same species that interact in both space and time. Questions? All right. Now we've defined populations. And in population ecology, most questions boil down to, here's a population. Is it going to get bigger or is it going to get smaller? At what rate will the population grow? At what rate will the population decline? What is causing the population to increase in size? What is causing the population to decrease in size? That's usually what we're talking about in population ecology. And then if we're talking about community ecology, it's like, are those multiple populations going to be going up, down? How are they affecting one another, right? So when we're talking about populations growing or populations declining, it's standard. At this point, I've read like 5 different ecology textbooks, and in trying to teach different classes, and every single one breaks this down into density-dependent and density-independent factors. So if you've heard this before, it's because it's just accepted now that every ecology class, this is the first thing we teach about population ecology. All right? So there are factors that affect whether a population will increase in size or decrease in size, and those can be categorized as density-dependent factors, something like intra-specific competition, or density-independent factors, such as a hurricane, all right? The area is prone to disturbance, all right? There's fires all the time. So density-dependent factors, they become more of a pressure on the population, meaning that they reduce growth rates or maybe lead to population declines. They become more powerful as the density of the population increases. So the size of the the density of the population, all right, is really what's important, not necessarily the size, but we can get to that later, okay, will actually be affecting how much of a pressure there is. So intra-specific competition between individuals of the same species, that's going to become more of a pressure as the density goes up. Diseases, that's going to become more of a pressure as the density of the population goes up. All right? Third example, that's going to become more of a pressure as the population density goes up. All right? Density-independent factors, things that don't care about how densely packed the population is. All right? Usually our example is stochastic weather events, disturbances, but sometimes, right, density-independent, like a fire isn't always a density-independent factor, because what if we're talking about a species like eucalyptus that is prone to burning and increasing the intensity of fires? Now, is fire a density-dependent factor because when there's a higher density, fires are more likely? I would accept that statement, and that seems like a pretty darn good argument. So whenever you set up a big dichotomy like this with 2 massive bins that you're trying to put all of ecology into, all right, there's going to be overlap, things that don't fit into the categories. Because the categories are artificial, we constructed them to help organize the complexity of nature, and it doesn't always work. Anyway, question?
That was my thought on the last slide on the different understandings of what a population is.
Yeah.
Like I feel like people switch in and out of those when talking about that.
I love that. That's so accurate. People switch between these definitions all the time. And in the common, and in the common vernacular, like we all just accept, I think that we usually accept when we're talking to people, like, okay, we might have a slightly different understanding of what we mean when we say species, you know, or population, or diversity, or something like that. But we have a certain general understanding that we are all working from, you know, and it's the same thing. When I say population, you know, I mean a big group of something. You know, that's what we all come to the table with, probably. But there can be differences in how you define the populations, and that's very important in how you set up your models and how you set up your frameworks. Is that a question?
No, I was just thinking to myself that thankfully population and diversity aren't one of those science terms that different disciplines use and it means something totally different. It's like the general idea is the same. It's the specifics that are different.
You know, I would say that works with population, but when we start to talk more and more about diversity, you're going to see just how much that can change, you know? Like people say it's a highly diverse community, and then we have all these assumptions about what that means, and they can be entirely wrong.
There's a reason we usually modify diversity.
Yeah, and the ecologist, probably of any field that you're going to encounter here, the ecologist focuses on the phrase diversity more than anybody else, at least as far as, like ecological diversity, community diversity, right? Good stuff. Genetic diversity. Yeah, this all matters. Yeah. Oh, this is great. So it's frustrating that these dichotomies and these categories don't hold 100% true, but we also need to accept it as just a given in science. And you just approach every time somebody gives you like 6 categories and tells you that all of reality fits into these 6 categories, just know that they're wrong. There's always going to be overlap. And they're coming at it from a very mechanistic, like oversimplified viewpoint. But that oversimplified viewpoint lets us make predictions, and if it makes accurate predictions, then it works for that scale. I know I'm getting off on a soapbox. I'm sorry. I just get really excited whenever I can try to get you all to think about that, Anyways, let's keep moving forward. Yeah, listen, does the density of the population change how important the factor is in regulating the population size? Question. Totally. So you've got a population of mice, right? There's 100 mice in a field, in the same field, or there's 10,000 mice in the same field. The fire happens regardless of how many mice there are. So the density isn't causing the disturbance to happen. Now, maybe the mouse population is changing the plant community and encouraging wildfire. Now it all of a sudden became density dependent. A better example might be a hurricane. An earthquake. An earthquake's great. Yeah, the plant or the animal community is not going to affect how likely it is that an earthquake's happening. Or how strong the earthquake is. Or like a volcanic eruption. Absolutely. Yeah. Solid?
So a density-dependent factor would be the animal and plant population impacts it?
Yeah, so competition, right? Let's go with these bunch of mice. There's 10 mice in this room right now. Did you know that? You eat 10 mice in a year in your sleep. Did you know that? That's a fact. All right, there's 10 mice in this room. There's a big bag of grain. All right, the population is going to keep growing and keep growing. Now there's 1,000 mice in the room and there's one big bag of grain. Those mice are going to be competing with one another for the grain. So competition is going to start to affect population growth rate. So it becomes more of a pressure as the population becomes more densely packed. Does that make sense? And the same thing with disease. If there's 10 mice in here, then there might not be that much disease pressure. Once there's 10,000 mice in here and they're all squirming over each other and they've got competitions, they've got open wounds and all this stuff, now disease begins to become a major pressure, which reduces population growth rates, maybe making those population growth rates become negative values. Reasonable enough? Awesome. Okay, let's move on. Here we go. So density dependent, density independent. All right, let's start to talk about how we deal with plant populations, because for many of you, you've probably approached population ecology from an animal viewpoint. And in the animal viewpoint, we usually talk about the number of individuals in a given area, and that's the density. But animals can move during their lifespan, and most plants can't, okay? So plants have what we call a sessile nature, where they're stuck in a given location, and so that means that density is really important, all right? Because it can't really just adjust. You can't have 50 mice in that corner so they just spread out. You can't have 50 redwoods in that corner and so the redwoods just walk away. You see what I'm trying to say? Solid. So, yeah. So density, sometimes I need to refresh people on this one, and that's totally okay. It's the number of individuals per unit area. So if we know like the number of redwoods per square meter, and then we know that a forest is 1,000 square meters, we could estimate the population of that entire forest by multiplying the number per square meter times 1,000 square meters in the forest. My hope is that when I said that, it was just like, yeah, obviously, of course. If that's not the case, then talk to me about it, because density is a thing that you got to get comfortable with, and scaling up densities to larger areas. Local density is important for plants because they can't move. So how's that feel? Everybody understand what I'm saying there? Sick. We're doing great. Let's start to talk about modeling plant populations. Okay? So, as with any ecology course, we first introduce this sort of functional model right here, where it's mainly designed to just lay out in a digestible way what's affecting population sizes. So let's take a look right here. N sub t plus 1. What does any of that mean? Well, n indicates the number of individuals. So n sub t right here is the number of individuals at a given time point. N sub t plus 1 is the number of individuals at a future time point. We're talking about plants now, so we're going to be talking mainly about geometric growth models, which are different from exponential growth models. Something that you'll have to refresh on if you end up going down the population community ecology route, because you use them at different times. Here's what I'm just going to try to lay this out for you, right? Plants tend to reproduce in pulses. Each season, they have a reproductive bout across that population, right? They go through their life cycle. They all flower at around the same time because they all need to pollinate at around the same time. They reproduce in pulses. That's something that we model with geometric growth rates. Some individuals reproduce constantly throughout the year, like cockroaches, so they follow more of an exponential growth where they're constantly reproducing. Question?
I was going to ask just for in terms of births, right? For plants, is that quantified, I guess, by the youngest a plant is that an ecologist doing a survey can identify it?
Great question. And whenever there's a great question, you know what my answer is going to be? No, I know this. It depends. It depends on who you ask. All right? So usually we talk about it as seed production, right? And a seed is determined as a new individual because it has distinct genetics, all right? But seed survival rates are incredibly low. So it can be pretty inaccurate, all right? So oftentimes what we'll do is we will quantify the number of seedlings, because when they are seedlings, they're now self-supporting organisms, right? They've gone through germination, they're carrying out photoautotrophy, so they can grow on their own, and they have passed through the biggest bottleneck. But it's going to depend on who's doing the population modeling. So what we do, and we'll talk about this a little later, is we break the plant's life cycle into stages, like the seed stage, the seedling stage, and then stage 2 or stage 1, which will be 1 year later. And then we'll say we built the model by surveying individuals that were at stage 1 or later. You see what I'm saying? So it's all the model designers get to decide, because it's really not very accurate to be like, well, the population's growing so rapidly because they're making 10,000 seeds, but then the germination rate is 0.1. Yeah, absolutely. Or just seed survival rate is that low because so many get eaten, they get attacked by fungi, bacteria, whatever happens. All right, functional model. Here we go. A poorly timed drink, but it was refreshing nonetheless. The population at some time, the population at some later time. Usually this is going to be the population in 2019, and then the population in 2020. All right? Because plants tend to follow that year-to-year cycle. But the t value can be different depending on what you're modeling. I know it could feel like a very silly thing what I'm telling you here, but it's important. What if you're doing bacteriology? They're not you don't want to know how the population changes from year to year. You want to know how it changes from week to week or from day to day. Anyway, so in this case, let's keep it really simple. N sub t is 2025, current year. N sub t plus 1 is 2026. This is a population of redwoods. The population of redwoods in 2026 will be positively impacted by the number of births throughout the year, negatively impacted by the number of deaths throughout the year, positively impacted by the number of immigration events throughout the year, and negatively impacted by the number of emigration events throughout the year. So this is immigration with an I, that means individuals coming into the population. This is emigration with an E, individuals leaving the population. It's usually, in plant ecology, seeds. All right? Yes?
Okay, that actually explains the emigration sense. That's
Yeah, how do they do it? Totally. So yeah, remember, the plant populations move, but it's all through their reproductive propagules. That was meant to be one of my main takeaways from previous couple lectures, but I didn't hit it as hard as I wish I had, you know? So there are immigration and emigration events, but it's through movement of seeds. And now you can ask, well, but what about if the model doesn't consider seeds as new individuals? And then I say, yeah, you got me, all right? But this is meant to be a very generalized model that works for any population. Births make the population increase, immigration events make the population increase, deaths make the population decrease, emigration events make the population decrease. Reasonable? Functional model, all right? Solid. There's a population size, immigration, emigration, death, birth. The arrows that are leading in make the population get bigger. Question?
Births are seeds, though, right?
It depends on who you're talking to in plant ecology, right? A seed is a new individual, so yeah, usually, but if you're trying to model the population realistically, if germination rates are very low, if seed survival is very low, you might be considering only a seedling as a new individual, all right? You could absolutely that's a great point. The births could be all the seeds, and there's a massive die out of those seeds because most seeds don't make it, especially if you're an R-selected species, right? Reasonable enough? Awesome. Okay, cool. And the key thing is population ecology, all right? It's probably what you're gonna study. You're gonna need to take a population ecology class, and then you're gonna need to go to graduate school and learn about specific species and how their populations are specifically modeled in order to really understand population modeling, because different species use different models. It's great stuff. Question?
To differentiate births and immigration, do you look at the genetics to see if that seedling came from a different population?
You can, yeah. Yeah, you absolutely can.
Because otherwise you just go, oh, it's a seedling.
Yeah.
Is it a birth or is it an immigrant?
Absolutely. So here's the thing, right? In functional models like this, it's not always important that we can attach values to these variables. This functional model is meant more to communicate this principle than for you to use it as an actual model, right? Because you can't measure these things. As you're saying, how do I measure immigration events? Do I have to take each seed and then sequence it? Isn't that gonna destroy the seed? Can I do that non-destructively and then put it back? How do I do that, right? And that's a really good point. So that's why these functional models are oftentimes valuable as learning tools, but then we need complex models where we can actually measure the variables. It's a really, really good point. I love it. Satisfying answer?
Yeah, so we would essentially be looking at them more as births because it's easier to look at it as immigration.
Probably.
If you're doing it in a simple model.
Exactly.
If you do it more complicated.
And usually our plant population models are really simple ones, are just gonna be you count. And you don't consider whether it was an immigration event or a birth from that population, and you're just counting from year to year. And then you just calculate a lambda value, and life is easy, you know? Yeah. Cool. We're good? All right. Solid. So yeah, some of these questions of like, what's a birth? All right. Is it a birth when the seed is mature? Or is it a birth when the seed is germinating? Like all of these are really reasonable questions, and it's all going to depend. All right. Genets versus ramets, just a quick reminder. All right. People might be concerned about the genetics, okay? Because oftentimes you're worried about, this comes back to our discussion of diversity that we had, the very brief one earlier, about like genetic diversity. So a birth is often considered as a genetically distinct individual, but do we consider a clonally produced individual as a birth if a new ramet gets produced? Or is it only a birth when it's a new genet? These are questions with the answer that I keep using. It depends. It just depends on whose model you're using, who you're talking to. So you need to be asking these questions when you're reading a population ecology paper and they're like, oh, the birth rate was this. And you're like, wait a second. What are you defining as a birth? You need to know that, in plant ecology, because that's going to differ. Anyway, good. Seeds are the mobile parts. Sometimes some people will model pollen flow, okay, pollen movement, because pollen grains are technically a new plant. They're just a haploid plant that's very short-lived. It's the gametophyte. It's just incredibly reduced. So is the gametophyte a new individual? It's genetically distinct from its parent. And we're concerned with the flow of genetics. It's not going to grow it on its own. Yeah. And how do you quantify pollen flow? It's kind of tricky. So you just wave a net around in the air. So, sometimes a model might even be developed where they talk about pollen grains, the gametophytes, as new individuals. But all to say, population ecology, maybe you just want to study animals. Because plants seem easier because they don't move around, but geez, this is frustrating. So we simplify it, all right? Oftentimes when you're talking about population growth, population decline, you'll use this lambda value, which is your fixed growth rate, all right? Fixed growth rate, you can just get it from surveys. So you get let's say you want to know what the fixed growth rate was for the year of 2024 to 2025. Well, all you have to do is do a population survey in 2024 and a population survey in 2025, and take the population in 2025 and divide it by the population in 2024. All right? So you've kind of gotten and now all you have to do is define, listen, we measured fixed growth rate. We only counted the individuals that had 3 leaves or more, or were larger in their diameter than 1 centimeter. All right, you define what the individuals are, and then you present the data, and this tells whoever's reading it, oh, the population was going up, or the population was going down. And it can let you project into the future, all right, a little bit, which is nice. You kind of like say, oh, well, next year, if the fixed growth rate was 1.1, and I want to predict what the population's going to be next year, I just take the population this year and I multiply it by 1.1. All right, the population's increasing by 110% year to year, and so I'm saying that it's going to be 110% of what it was this year next year. Nothing too wild, right? You have a paper that's assigned in one of these weeks, and it talks about lambda values like this. So check this out. How do we use it for projecting far into the future? Well, don't, first of all, because there's going to be these density-dependent factors that come into play, and the population isn't just going to infinitely increase. But you can project a little bit into the future, and just by taking the population at an individual time and multiplying it by the fixed growth rate raised to the number of growth of reproductive intervals, whether it's every 3 months or every year. All right. But what that predicts, those models always predict this. That the population just goes, keeps going up and up and up and up and up, and there's nothing stopping it. But you know that there are these density-dependent factors that are involved in population regulation, in population size regulation. So once it hits a critical density, it's going to level off. All of this, okay, listen, we use geometric growth rates, geometric exponential growth rates. I don't want you to stress about this too much, because what I want to do is sort of push past it into a more functional model, because in ecology, we always start with these models and then say, but this never happens, you know, because the populations eventually level off. The only time that you get true like exponential growth or limitless geometric growth is in like a petri dish or a liquid culture of bacteria, where you just keep providing more liquid and you keep providing more nutrients, so there's never anything limiting and they can just keep reproducing. But if we're talking about a plant population or an animal population, the growth curve tends to go like this and follow the logistic growth curve. It's a sigmoidal curve, it's S-shaped. We talked about it already with the R versus K selected species, and this utilizes, it's a little bit small. This utilizes a more useful model. So we're kind of just shooting right past the exponential and the geometric growth rate models and instead using a more useful logistic model of population growth. Okay? So population reaches its carrying capacity, and then it fluctuates around it. Usually, people will say that the carrying capacity is determined via intraspecific competition. Okay? Because this model here, it only considers a single species, the number of individuals of a single species, the carrying capacity of that species, and the growth rate, the max growth rate of that species. So if the model doesn't have any terms for other species or for stochastic events, right, then this can only be related to density-dependent factors like intraspecific competition or disease. But we try to bundle all of these things that can limit a population size into one term, the carrying capacity. Determining carrying capacities is its own, its own subtle art, all right, which usually involves surveys. And you find historical carrying capacities and you use those. End of the day, that's why so many ecologists end up, you're just surveying all the time. And it's always justifiable, because you're like, well, we need that data for generating these models, because the actual variables are too complicated. Anyway, population, right? Here's the model that you actually have to know. And I actually want you to learn this equation, okay? If I ask you questions about it on an exam, the equation will always be provided, all right? If you need to use a calculator on an exam, I will give you plenty of heads up that you need the calculator for that exam, all right? The math's never going to get really more complicated than this logistic model of population growth, though. We do life tables here, which can be tricky, but I'm going to give you tons of examples of them. Where are they? But I'm going to give you tons of examples of them, okay? And we're going to do very simple life tables. I just don't want you to leave a plant ecology course never having encountered these. Okay. There we go. So let's talk about it. Let's talk about the logistic model of population growth, which I've been talking about in a lot of different courses, so I'm kind of fuzzy on how much of it we've talked about in plant ecology. We haven't talked about it yet. I'm seeing shaking heads. Cool. In that case, I'm going to start from the start. let's first define the variables in the equation. That's a great place to start. So, the logistic model of population growth gives us a rate of change. Delta indicates change. This dn, it just stands for delta, or the difference in number of individuals over difference in time. So the growth rate of the population. Cool so far? We're going to be calculating the growth rate of the population. You use this variable called R sub max. This variable is what we term the intrinsic rate of increase, okay, for the population. So what's the growth rate, R, under ideal conditions? How rapidly can the population grow if there's nothing limiting its growth? Does that make sense? Awesome. You multiply that by the number of individuals. Now check this out, right? If you ignore the parenthetical for a second, if you wanted to predict a population's growth under ideal conditions, you would just need to multiply your R sub max times the number of individuals, and that's going to give you the number of individuals next year, right? Just like with that lambda value that I talked about before, okay? The only difference is this R sub max value is the intrinsic rate of population growth, the maximum rate that it can reproduce at. So if it's 2, that means the population's doubling every year. And if there's nothing limiting the population's growth, it's going to double every year. Now, we incorporate this additional parenthetical, and this parenthetical has your carrying capacity in it, okay? So n is still the number of individuals in the population. K is now the carrying capacity, or the max population size that the environment can sustain. All right, we cool so far? Oh, was that helpful? Slide 15, please. Okay. Oh my gosh. So check it out. If we look at this curve, as the population nears carrying capacity, its growth slows. All right, and then it levels off at the carrying capacity. So what actually happens as n gets closer to the carrying capacity mathematically? All right, if n is equal to k, let's say there's a carrying capacity of 1,000 and the population's 1,000, well, 1,000 divided by 1,000 is 1. 1 minus 1 equals 0. So the population's growth rate is 0, and it levels off. That's why the model makes this prediction, all right? If n is less than n is less than k, there's 900 individuals, there's a carrying capacity of 1,000, well, then that means that this term here is 0.9. 1 minus 0.9 is 0.1. So the population is effectively growing at 10% of its maximum growth rate. All right? Because it's at 90% of its carrying capacity. And that's the prediction of the logistic growth model. And if this value, if it reaches 1,100, okay, and the population, the carrying capacity is 1,000, well, now this is 1.1. All right, 1 minus 1.1 is negative 0.1. So now the population is declining at 10% of its maximum growth rate. And that's how the logistic model works. That's where we'll come back to on Thursday. I stole a minute of your time. I'm so sorry. Have a good break. Weekend's coming. See you on Tuesday. It's going to be a hoot and a holler. We're going to do life tables. Everybody loves them. If you need to get your exam from me and you've got a few minutes right now, you can totally come tag along with me to my office. All right.